colony description. Bacteria can be grown on an agar plate, which is called culturing. When bacteria are grown on an agar plate, they consume the nutrients within the medium. Each single bacterial cell replicates many times to generate millions of cells that form a single bacterial colony on the plate. The appearance of these colonies is termed colony morphology. Describing colony morphology is the first part of identifying and characterizing a bacterial culture, because individual bacterial species often form colonies of characteristic size and appearance. The four main characteristics we use to describe bacterial colonies are size, shape, colour, and any other distinguishing features, such as whether the colony surface is glossy, slimy, smooth, or dry. Here we have bacterial colonies growing on a blood agar plate. It's called blood agar because it contains horse blood. Blood agar is one of the most routinely used agars as it contains many nutrients suitable for even the most fastidious bacteria. Note that there are two colony types growing on this blood agar plate. The colony morphology of the smaller colony would be described as white, round, and 2 mm in diameter. The second colony type would be described as grey, of an irregular shape and about 3 to 6 mm in diameter. Some bacteria also cause lysis of the red blood cells in the agar. This is described as hemolysis. The best way to check for hemolysis is to hold the plate up to the light, like so. Look for clearing of the opaque red blood cells in the zone surrounding each colony. Note the plate on the left has complete degradation of the red blood cells. This is called beta hemolysis, while the plate on the right has only partial degradation of red blood cells. This is called alpha hemolysis. Note the greenish tinge around these bacterial colonies.
We're going to flame the lip of the test tube to sterilize anything in there. Bring our loop down to the bottom and then just streak up. The idea that our inoculum, our bacteria, is going to inoculate along that slant of the tube. Flame the lip of the tube again before closing it, and once again flame your loop.
Now growth on agar slant. In slants, when you inoculate through your wire loop, inoculating loop, so there is also various type of colony formation. But their colony formation is different from plates. In plates, they grow on grow as a certain, and in certain types, and in test tubes they grow as certain types. In agar slants, there are filiform shapes, tree-like shapes, beaded shape, then diffuse or spreading shape, and rhizole shape. I am zooming it so you people can easily see that. Then this is the filiform shape. Okay, this is tree-like shape. There will be tree appearance. And this is bead. Bead means there is small bead-like appearance will be there. Then there is a diffuse, and there is a rhizoid is also there.
As always, you should work close to the Bunsen flame and avoid putting any lids down on the bench as they might become contaminated. The broth container is first labelled with the information previously described and its top loosened. A cool sterile loop is used to pick off a few single colonies from a pure culture grown on a solid medium. The other hand picks up the bottle and its lid is removed. Once open, the bottle neck is flamed. This flaming draws any aerosols in the bottle into the Bunsen burner flame. The bottle is slightly tilted and the inoculum on the loop is rubbed onto the inside of the glass bottle, so that when the bottle is upright, the inoculum is submerged. The bottleneck is then flamed once more and the lid is replaced. Finally, the loop is sterilised, so it is safe to leave on the bench. After appropriate incubation, a broth will appear cloudy or turbid. If a broth remains transparent, it means that the microorganism hasn't grown.
This demonstration is to show you how to inoculate a slant. A slant is culture media that contains agar that's placed in a tube that has a screw cap. The liquid, the culture media, is autoclaved. When it comes out of the autoclave, it's placed on boards so that it solidifies and we have a slanted surface. I'm going to be inoculating the slant with bacteria that I have in another slant. To do so, take your inoculating loop, sterilize the loop, the wire, the bottom of the handle. Uncap the tube, take the inoculating loop, touch the surface of the colony, replace the cap. Open up the cap of the tube that you'd like to inoculate, take the inoculating loop, put it on the bottom of the slant, on the surface, streak it back and forth as you go up the tube. Replace the cap. Sterilize the loop. Place your tube in the incubator. In the next class period, you should find growth throughout the tube.